I
Yeah.
think several things. First, I should say that thank you all, the speakers throughout the sessions. And what we have planned for this last session is the four organizers, Herbie Levine, Phil Sharp, Rick Young will be joined by, and myself will be joined by Krastan Blagoev from the National Science Foundation. And we do not intend this discussion to be a series of pontifications from us, but rather to really have this as a free-flowing discussion, which we will seed by each one of us saying a couple of sentences about the meeting and the future, and then it will just be a free-flowing discussion. So let me start, since I'm already talking, and say that I thought that there were all the talks were great, but some that stood out in my mind just randomly right now is Mike Rosen's talk in the beginning. There are many other lovely talks, such as Alexandra Zudovska's talk and Dave Termlai's talk, and I thought all the talks in the RNA session were just great, and also many talks from the Rice group. And finally, I'd like to talk about Gita's talk, because this was perfect meeting talk to end this meeting with. I mean, it was a spectacular talk in my view on every level. And because of things that I'm thinking about, and I also think people will agree that this is the future, what her talk encapsulated was the importance of underlying free energy landscapes that promote phase separation, that rely on electrostatics, or at least partially dependent on it, and that they occur on a polymeric background. And all of these processes are driven far from equilibrium. And I think that's, in my view, is where, as a somewhat new person in this field, feel that's the direction that will define and help understand the real processes. So with that, I will stop. I probably will have almost nothing to say other than to help moderate the session in case somebody acts unruly. So that's my job from here on. So Phil, I saw that you were on. Would you like to say some things just to seed the discussions?
I thought the symposium was excellent, and I thank all the speakers and the participants. It came together pretty quickly, and I have been impressed by the range of topics and the speakers who've been engaged. I also particularly liked the last presentation by Gita, and thought it raised a lot of questions, even though it is a pretty defined system, as she points out, and we will have to begin to explore the diversity even among HP1 proteins and the nature of the condensates they will form. I was interested in the question of, would an RNA generated in the midst of that condensate with electrostatic principles as its major or contributing part of it, would an RNA actually move from one of those domains of a chromosome to another? Because I think then we may see some interesting properties of that condensate in terms of its engagement with RNA. I chaired the session on RNA, which really also raised in X-Cis is silencing a number of questions about how RNA organizes proteins that are involved in silencing various regions of the chromosome. And I think the integration of the properties of proteins with DNA and RNA is going to be a field rich with complexity, rich with specificity, but and also rich with implications for how we can approach biological problems and even interpret phenomena that we at present can't. So a very rich topic, a very rich field undergoing a transformation that will be, as I said at the beginning in the brief comments, that is introducing the element of dynamics into cell biology in ways we've not been able to do before, and integrating physics with a cell and molecular biology in ways that the cell biologist side isn't frequently been able to do. So a very exciting symposium.
Maybe we'll go to Herbie next.
Thanks. So this is a very interesting symposium, partially because I'm an outsider to this field. I actually don't work on chromatin. So I have some comments, I guess, in two directions. One from sort of a basic physics direction, which I think the two bookend talks that Arup already mentioned, the one by Rosen in the beginning and the one we just heard, were really great at sort of setting up the question of, we know that we can make really nice in vitro systems that look like complex versions of polymer physics, phase separation, that really look like things we can get a handle on, we can understand quantitatively. We all love that. And then the question becomes, how much do they relate to the in vivo systems that we actually care about from the point of view of biology? And I think the two talks, sort of, the one at the beginning, the one at the end, sort of brought that question to the fore in a brilliant way. And so I think that's one of the directions we have to go in. I mean, it's very nice to do polymer physics, but we really want to do biology in a sense at the end of the day. So in that regard, there are many questions. I guess I still don't quite understand in a biological context when I'm supposed to think of some structure as a liquid condensate or a liquid phase separated region versus a protein, multi-protein complex. So if I think of the just to remove it from the realm of chromatin and the nucleus, if I think of the postsynaptic density as a region with many, many different types of proteins, it's not one protein, it's not a simple model system, it's a complex system. Am I supposed to think of that as a liquid condensate? Am I supposed to think of that as a organized multi-protein complex where each individual protein has a specific spatial location and a specific set of binding partners? I don't know the answer, but I think that's a sort of fundamental question that I still think about whenever I hear talks about this various subject. Connected to that is the issue of does the non-equilibrium nature change some of the features? For example, I think Arup mentioned, and I think the talk from Johns Hopkins on the issue of to what extent coarsening behavior that we would expect in an equilibrium phase separation system, either by cluster aggregation or by Oswald ripening or whatever, to what extent those are somehow precluded or gotten rid of by non-equilibrium effects, I think is still an interesting question which some people in the room have worked on. So the other set of comments has to do with, there are many talks that somewhere along the way in the talk said, we're really interested in the connection of this structure, of this dynamics to gene expression. And then that's the sort of final statement in that regard. Now I'm not blaming the talks. I mean, you first probably need to understand more about the structural elements at different scales before you can really understand in detail how they affect gene regulation. But my main interest these days is on sort of dynamic systems biology, how networks of genes and networks of RNAs interact with each other dynamically to actually enable cell differentiation, to actually enable changing phenotype in a cell under stress. And I think this field eventually has a lot to say about those processes, but it's not quite there yet. I mean, I still think, gene expression is being used as it seems to me, as a static indicator of, well, we can look at RNA-seq and we can find regions where the gene is highly expressed, and that correlates well with, some type of domain structure as seen in high C. We can obviously know the separation between heterochromatin and euchromatin. Those are all, in some sense, static indicators of the correlation between genomic structures at different scales and gene expression patterns. It's not yet at the level of, how dynamically do I have a pioneer factor? It comes in, it opens up chromatin, it calls in the right factors, it dynamically gives rise to a change. How does the interaction of the chromatin degree of freedom together with the sort of transcriptional network degree of freedom actually work to limit the possibilities of state change, whatever? Those are the questions I'm ultimately interested in. And I think, you know, when this meeting goes on, I think Creston will say that we want this meeting to be an ongoing event. I would hope that we would see more of an integration of those two fields together in terms of dynamic gene regulation and how that's directly affected by these chromatin structural elements at these different scales. So with that, I thought it was fantastic. I certainly get to learn a lot because I knew so little at the beginning. And I thank very much Arup for doing the bulk of the organizing of the effort. Being on an organizing committee with Arup is a very, you get some credit and you do no work. So it's great. And with that, I'll turn it back over to him.
Well, maybe we'll go with Rick first, and then Creston will not get the last word, but the last word for this first round of comments.
Well, with respect to the talks, the striking thing to me was the level of sophistication and polish of the talks from the fellows. I think you all should be commended for the work that you did with those talks, especially in the context of Zoom. You know, it strikes me this meeting was about nuclear processes, and within the last few years, we've come to understand that most nuclear processes are compartmentalized in condensates. And the remarkable feature of these condensates is that they're encompassing regulatory processes that have historically been the subject of detailed genetic, biochemical, and structural studies. So we know a lot about underlying molecular mechanisms, but now we have this additional regulatory level of condensate behavior. And so I think what's interesting is we're integrating a conceptual understanding of a historical, molecular, biochemical, genetic nature and a new biophysical nature that I think is leading to some fundamental changes in the way biologists and even physician scientists understand how human biology works. So I think we're now in an area where physicists and biologists, not to mention chemists and folks who work on artificial intelligence, really have to collaborate to gain deep insights. And I just leave you with 4 questions for discussion. What are the components of these condensates? We know RNA plays important roles. In the nucleus, DNA is often a component. Proteins are certainly components. But we don't really have a catalog of the components and condensates. What are the features of molecules that cause them to partition selectively into those specific condensates? We have for FUS and a few other proteins insights into what has been termed the molecular grammar, the amino acid side chains, the stickers and spacers, the ways that we currently think about partitioning behaviors. We need much more understanding of those features. What's that physicochemical environment look like inside a condensate? How does that differ from that outside? If drugs, for example, become concentrated in condensates where their targets lie, is there a different kind of pharmacodynamics we need to study? And finally, what are the non-equilibrium events that drive dynamic formation and dissolution of these condensates? I think the answers to these questions are well within the command of many of the experimental and theoretical systems that have been presented here. So I thought they might be worth further discussion.
So thanks, Rick. Creston?
Yeah, so in terms of the science, I think this was a fantastic symposium, and thank you, Arup, and the organizers for organizing this meeting. I want to thank also Sarah, Kelly, and the team at MIT for excellent performance and almost completely flawless meeting. In terms of the science, so I want to go a little bit back to the late 20s, early 30s of last century, when condensed matter physics was starting to become a field. And condensed matter physics, starting, say, with the Ising model, completely developed new physics tools and language, which didn't exist in the physics community before that. I think that we are still in the very early stages of doing this in this field. We are using, and I'm guilty of that too, we are using models that we have developed in other contexts for other systems, as Herbie said, you know, polymer physics. We need to move on theoretically and develop concepts that actually emerge from the system that we are studying. There will be analogies, of course, and there will be things that probably we already know from other systems. But I expect that the integration of non-equilibrium physics with networks that are functional networks will lead to actually new language and new physics. We are loosely speaking about condensates. Probably in different contexts, these are different condensates, and whether they're condensates at all is still a question for me, at least. In the P granules, there is a lot of work, as you know, by Cliff and by other people, and they did show to a large extent that these are condensates, that they actually behave like a liquid, that the droplets behave like a liquid. They dissolve and organize. We still don't know what drives this P granule formation. But these are the questions we want to understand. We want to understand in different contexts, what is the common and what is the difference between these organelles. And so I see the future, in fact, going in that direction, more theory, working with the wonderful experimental systems that we have. It's not easy to study in the cell. Again, Cliff and others have developed now multiple model systems where you can optically induce those droplets and dissolve them. But again, what is the role of ATP? Some of these things might, as Herbie said, might be assemblies and not actually, might be aggregates, not condensate, not liquid condensates. They might have to be dissolved, as you guys have shown at MIT, by ATP-driven processes. There is a recent paper by Cliff, again, showing that the phase diagram, the saturation concentration depends on the concentration, which was unexpected for these systems. So there are a number of theoretical questions that we do need to understand. And that's why I would really, as Sarup said, would like this to be an annual event. We will have, of course, a meeting in the spring, and we should discuss with the organizers and with maybe broader range of people, do we need to have another meeting next summer in addition to the March or April meeting, or do we want to skip 1 year and go for 2022? But certainly the interest is huge. People are interested in these problems. And so I'm very excited. And from my point of view from NSF, at NSF, we're very excited together with my colleagues in MCB of this field. This field is really, really cutting edge field at the interface of physics and biology. And I think that we really have a chance in the next decade to understand these processes and to actually make cellular biology more quantitative than it's now. So again, thank you very much, and I'll give it to Arup.
So I think what would be good now for a few minutes is to have people who are on the call to contribute to the discussion. And I personally, and I'm not forbidding others, but I see the first hand. The first hand is from a person with lots of gray hair. I would like to see some people participate in this discussion who are not in that category. So having said that, I will let Jose Onachek speak. But I do encourage younger people to contribute to the discussion because you are the future.
Thank you, Arup. Thank you for the organization. It was a great meeting. Thanks, Creston. I think that's a hot topic. We are all are very, very excited about working with you to have to make this meeting annually. And we thank MCB and the Physics of Living System at the NSF to support this idea. I think the meeting was fantastic. I think there was a comment by the speakers about some of the talks by the senior people, but I really enjoyed the talks by the young people. Like Arup was saying, I think they really were short talks, but they really brought to the front a lot of the new things. I want to bring a couple points here One is that basically both theoretical and experimental, we keep talking about structure and function, but structure is a very complicated thing as we come to this sort of chromatin. Basically, all have been common. We did that, but several other people working on it. The idea is you have an ensemble of structures, is a structural heterogeneous ensemble, but also dynamic ensemble of structures. And it's very, very interesting to understand how to differentiate this ensemble of a single cell type from ensembles of different cell types and try to figure out how, if you're looking at two particular structures, it's very easy to differentiate them. But you have ensembles, basic, which are the proper coordinates. We saw a lot of discussion today about that. Tells you how to identify a cell type based on its structure, not based on it. And I think what you're very interested in is the connection between all the way of epigenetics and structure and dynamics become a very interesting question. And the question of ensembles, what is just fluctuation of a single cell, heterogeneity of a single cell versus difference between different cell types or different organisms. There's a lot of diversity here to be explored. Second, I want to comment what Creston said. I think you're using the word phase transition and condensates in a very loose way. And I think we really need to quantify that. In which of these cases, these are just dynamic effects that we look at some fluctuation when you have something where the direct connection to phase transition becomes important. I think particularly have finite systems and we are trying to say, is that a liquid or that's not a liquid? I think you're going to have a lot of different phases of matter come here that they're not can be classified with these very simple descriptions we have. I think this problem makes this problem very interesting. So in a certain way, I like what Phil and Rick Young were saying about this sort of dynamical new cell biology. There's an interesting, very new biologist coming from it. A lot of understanding how these things work. But the counterpart of it, and that's also goes to the comment that Creston made, there's a lot of new physics that's coming from it. So I think it's a win-win situation, basically. We are going to have to learn new basic physics to come from it. I just put the idea on phase transition cell type. So I think it's going to be a rich field, not a field just to understand the life science, but also understand this sort of physics of complex systems and the entire thing of non-equilibrium system and networks coming together. So we really need the young people, and I want to thank for the meeting. I'm looking forward for the new version in March and future extensions of this meeting. Thank you.
Yeah, so I would like to hear from young people. I might just comment on the last thing that Jose said. I don't want to debate about it, but just to say that, you know, the new physics will come from the kinds of questions that I brought up in my very brief remarks, which were connected to some of the issues that Gita brought up. And some of that is beginning to emerge. In any case, young people, we need commentary from young people. Don't be shy, please.
Arup, you have to define young before we hear from them.
Young people are all people younger than me.
Okay, so everybody, half of the meeting is younger than you.
And so everybody who's spoken so far is not, except you, Creston, is older than me. So younger people now. Great, Zane Thornburg and Michel de Pierrot. So Zane first.
So this meeting was very exciting for me as a younger computational graduate student. It's just good to have something this focused on parameters and experimental techniques we care about. So I'm trying to learn more on eukaryotic systems right now. Right now I mainly work in bacteria, but I just find it exciting to have something this focused that delivers new experimental techniques, such as the ones from TJ Ha's group that we heard about. And then just the actual, when we do our simulations, we care a lot about maintaining our use of as many experimental parameters as we can. So the push for measurements of diffusion and affinities and reaction rate constants, those sorts of things. This meeting, I think, it seems like everyone pushes each other to do a better job at obtaining these measurements, which is very exciting for me on the computational side.
Thank you. That's, I think, an important comment. And we'll now go to Michel de Pierrot from Northeastern University.
Hi, so I am relatively young. I'm an assistant professor and I'm just starting my tenure track. So in the physics department. And I wanted to, okay, first I would like to thank the organizers. This is a great meeting and very, very interesting. And it was very interesting to see all the different facets of the problem. My personal interest and why I'm really excited about this field and I'm setting my lab fully in this domain is the new physics, because, and there's two things that are interesting to me to the new physics. First is the new physics is going to be a physics of somewhat liquid, somewhat semi-condensed. So it's the physics of really highly heterogeneous structural ensembles and how we connect that to function, as Jose was pointing out. But also, as Rick or Herbie were pointing, this physics needs to be a physics that is specific. It's not a physics of every protein. It's a physics of why a certain protein acts in a certain condensate or in a certain way. So I'm really interested in this sort of like moving physics to be more specific to biology. And I think there's a lot of challenges in that, both theoretical and computational. I think this is going to be really great few years ahead. And yeah, I would love to participate every single year. Thank you.
Thank you. Any more commentary and questions, discussion? I think people are getting tired after 3.5 days because I can watch people dropping off, but yeah, any further commentary from those who are on? Well, I don't see any further hands coming up or things sent to me by chat. So let me say the following in closing. As we've all commented, great meeting, very heterogeneous in terms of the areas covered, but all still integrated. And I'm glad you all will be able to participate across the world. We will definitely have the in-person meeting that we had planned, which will have, with one exception, a distinct set of speakers. And we hope to have that when conditions permit. Kostan pointed out that, and others have too, that they would like to have a meeting following that, either in the summer or the following year. What I can say for myself is that having played some role in organizing this video meeting and the one that will happen in person in the spring, I will not be organizer or the principal organizer of the next meeting. That I will tell you, but I am sure that others will step in to do that. So on that note, I'll bring this meeting to a close, and I thank you all for the participation, and it was great. Thanks all.
Thank you.
Thank you, Adam.